Welcome guys. In this topic we are going to discuss about the gluconeogenesis process. Now what is gluconeogenesis? Gluconeogenesis means we are going to make the glucose. Gluconeogenesis, we are going to make the glucose. Now why this word neo is used here is the reason for the fact that there are the non-carbohydrate molecules, that will be converted into glucose. So it is called as gluconeogenesis. Non-carbohydrate will convert into glucose. Say for example fatty acid, amino acid, they are the non-carbohydrate thing and we are converting them into glucose, so that is called as gluconeogenesis. This process occurs mainly in liver. The place is liver. So 90% of the total gluconeogenesis is going to occur in liver. Somewhat can occur in kidney also. So around 10% of the gluconeogenesis occurs in kidney also, but mostly is going to occur in liver. If they ask in the liver where, what is the organelle where it is going to occur, it is partly occurs in the mitochondria and partly occurs in cytoplasm. Partly in mitochondria, partly in cytoplasm. Means few reactions are going to occur in mitochondria, few are going to occur in cytoplasm. We will see that in the pathway that there are few reactions that are occurring in the mitochondria, some reactions are occurring in the cytoplasm. The next thing that is important is what are the non-carbohydrate substrate from which we can make the glucose. So what are the substrate of gluconeogenesis? What are the substrate of gluconeogenesis means what are the molecules by which we can make the glucose. So starting with the first one, the first substrate of gluconeogenesis that we will discuss is pyruvate. Pyruvate is not a carbohydrate. It is not a monosaccharide, disaccharide or polysaccharide. Pyruvate is not a carbohydrate. From pyruvate if you make glucose, yes, it is called as gluconeogenesis. The second is we will discuss from the lactic acid. How the lactic acid will convert into glucose. Then we will discuss about the various amino acid. Amino acid is the main source, or I can say the major substrate. The major substrate of gluconeogenesis is amino acid. Then we will discuss about the oat chain fatty acid, how the oat chain fatty acid will convert into glycerol. And lastly, we will talk about the glycerol, how the oat chain fatty acid will convert into glucose, and then we will talk about how the glycerol will convert into glucose. So these are the various substrates. Now whenever the question is asked that all of the following are substrate of gluconeogenesis except, there is one molecule by which we can never ever make glucose. There is one molecule by which we can never ever make glucose, and most of the time that is going to be there in the option. And the name of that molecule is acetyl CoA. So let's write down this negative point that acetyl CoA is not a substrate of gluconeogenesis. Is not a substrate of gluconeogenesis. We can never ever make glucose from the acetyl CoA. We can never ever make. Acetyl CoA is never ever a substrate of gluconeogenesis. You can make glucose from any of the molecule, but you can never make glucose from acetyl CoA. This is the statement that you need to remember. So now let's start with the first one, that how we can make glucose from pyruvate. So I am writing pyruvate to glucose. Pyruvate to glucose. So to make you understand the pyruvate to glucose, I want briefly to remind you the concept of glycolysis. If you remember, if you have seen the session of glycolysis, in that we have discussed that glycolysis consists of 10 steps, and out of 10 there are 3 irreversible steps are there, the first, last and the rate limiting one. This is what we have written. So what I can say is, if I just write the irreversible step of glycolysis, what I can say is, we have written that glucose, if I just write the irreversible step, the first was the irreversible, glucose converts into glucose 6-phosphate. That is the irreversible step in the glycolysis. Then the next step was reversible, converts into fructose 6-phosphate. Then the rate limiting step was there. This is irreversible, fructose converts into fructose 1,6-biphosphate. Then the further reactions were reversible till the last one. The last was phosphonyl pyruvate converts into pyruvate. Basically, these are the 3 irreversible steps are there in the glycolysis. These are the 3 irreversible steps. So let's highlight them with the yellow color. Let's highlight them with the yellow color. These are the 3 irreversible steps. So currently, why I'm discussing about the glycolysis here in the gluconeogenesis? This is the point because currently we are standing here. Currently we are standing here at the pyruvate molecule. Currently we are standing here at the pyruvate molecule, and we want to go back all the way up till the glucose. We want to go back all the way till up the glucose. We know that this process occurs inside the cytoplasm. This process of glycolysis occurs in the cytoplasm. So right now we are standing here and we want to ascend up and we want to reach all the way till glucose. So how many hurdles are going to be there? There will be 3 hurdles, and that is highlighted with the yellow color arrow. These are the 3 hurdles. If I am at pyruvate, I want to go back to glucose. These are the 3 hurdles that I have to bypass. So what I will do is I will take the alternate route and will bypass these hurdles. Means what? What I will do is pyruvate, I will take the alternate route and will reach here. Will reach here. Then I can use the same enzymes because these are the reversible step, will stuck here. Then again I will use the alternate route, will reach here. Then again the same enzyme, then stuck, alternate enzyme, and will reach here. So this is what we need to discuss basically in gluconeogenesis. What is the alternate steps? What are the alternate steps by which we will bypass these 3 hurdles? So let's understand these alternate steps by which we can bypass these hurdles. So currently we are currently here in the cytoplasm. Let's say this demarcation which I am making, it is for the mitochondria. This side is the mitochondria and outside to the mitochondria is the cytoplasm. Outside to the mitochondria is the cytoplasm. This is the demarcation that is there. Now this pyruvate will come inside the mitochondria. If I want to bypass the hurdle, the pyruvate will come in the mitochondria. Pyruvate is a 3-carbon molecule, we know. It will convert into a molecule that is of 4 carbon, that is oxaloacetate. It will convert into a 4-carbon molecule, that is oxaloacetate. Oxaloacetate is a 4-carbon molecule. 3 carbon converting into 4 carbon means we need to do a carboxylation reaction. So the name of the enzyme is pyruvate carboxylase. This pyruvate carboxylase requires energy as well as it requires vitamin B7. Vitamin B7. Vitamin B7, the name of the vitamin B7 is biotin, that helps in the carboxylation. Decarboxylation, vitamin B6. Here we are doing is carboxylation, that we require the B7. So we have got the oxaloacetate. Now this oxaloacetate will come in the cytoplasm. Oxaloacetate comes in the cytoplasm. Oxaloacetate is a 4-carbon molecule. When it comes via a shuttle. The name of the shuttle which is there, which is going to help here, it is called as the malate shuttle. With the help of malate shuttle, the oxaloacetate will come in the cytoplasm back. Then this oxaloacetate will try to convert back into phosphoenol pyruvate. So that we could have bypassed the first hurdle. See, we have taken the alternate route, we came inside and we are going like that. So that we could have bypassed the first hurdle. Now to convert 4 carbon into 3 carbon means what? We need to remove a carbon dioxide. Then oxaloacetate does not have the phosphate group, but if you see the phosphoenol pyruvate is having a phosphate group. So what we do is we bring ATP into the picture, we transfer a phosphate group and ATP will convert into ADP. So again we are transferring a phosphate group from the ATP. So the name of the enzyme is going to be kinase. There is one more thing that you are doing is you are removing the carbon dioxide, so it is going to be a decarboxylation reaction. So the name of enzyme is phosphoenol pyruvate, PEP, phosphoenol pyruvate, PEP, decarboxykinase. Because we are doing the decarboxylation and we are transferring the phosphate group, so the name of the enzyme should be phosphoenol pyruvate decarboxykinase. But it is a misnomer. Instead of decarboxy, what they say is, although we are doing decarboxylation, but in the literature it is written as phosphoenol pyruvate carboxykinase. It is a misnomer, but it is there and we have to remember it like that way only. that the name of the enzyme is phosphoenol pyruvate carboxykinase. You are going to remove a carbon dioxide, you are going to transfer a phosphate. So this is how we are going to bypass the first hurdle. You have taken the alternate route, you have went inside the mitochondria, then came out all the way and reached all the way till phosphoenol pyruvate. Now there is no problem. Now we can easily ascend up with the same enzymes we will ascend up, and we will stuck at this point. We will stuck at fructose 1,6 biphosphate. So again we have to use alternate route and we will reach here. To convert the fructose 1,6 biphosphate into fructose 6-phosphate, we will use the enzyme fructose 1,6 biphosphatase. We will use the enzyme fructose 1,6 biphosphatase. Once we will make the fructose 1,6 biphosphatase, then again we will use the same enzyme, we will stuck here because it was irreversible. So to convert the glucose 6-phosphate into glucose, we will use the enzyme glucose 6-phosphatase. So this is how we can convert all the way pyruvate. We can ascend up and we can reach all the way till glucose. There were 3 hurdles. To bypass these 3 hurdles, first we went inside the mitochondria, then came outside, and then ascend up, then stuck the alternate route, then ascend up, then stuck, then the alternate route. So this is how we can make glucose from the pyruvate, right? There were 3 hurdles. To bypass them, we have used 4 enzymes and 1 shuttle. 4 enzymes and 1 shuttle. So this is how the glucose can be made from the pyruvate. This is the first substrate of gluconeogenesis. Let's see the second substrate. The second substrate that we are going to discuss is lactic acid to glucose. Lactic acid to glucose. How it is going to be done? See, let's first understand how the lactic acid is going to be generated. Lactic acid is generated in a way that, for example, this is the skeletal muscle, and skeletal muscle requires glucose for the energy purpose. Let's say you went to the gym, you are doing heavy exercise, your muscle requires a lot of energy, so the muscle will burn the glucose in the form of glycolysis. So the product that you are going to get is pyruvate. Because you are doing this reaction so fast that there is no enough amount of oxygen is available, as I am saying that you are doing vigorous physical exercise. So what happens is this pyruvate will undergo anaerobic oxidation, anaerobic pathway, because that much amount of oxygen is not there continuously. So when you do the anaerobic glycolysis, you may have noticed that pyruvate converts into lactic acid. And this is how the lactic acid is generated in the muscles. And this is the molecule which basically leads to pain when you go to when you do any physical exercise, vigorous physical exercise, which is not routinely done by you, and that is not there in your routine, and you have done that for the first time. Now because of that, what happens is the excess lactic acid is going to be generated, and that is going to be accumulated in the muscle, right? And that is why you may have seen that after doing the gym, when you go to the gym, your muscles seems to be more, we can say, swollen up. They looks much, we can say, healthier or swollen. So that is because of the accumulation of the lactic acid. Now what is going to happen with this lactic acid? See, this lactic acid will slowly will drain out, and via the blood, these lactic acid molecules will drain out. Again, your the muscle which was looking very swollen, which was looking heavy, which was looking bulky. Now as you will come from the gym, let's say you went to the gym in the morning, you did vigorous exercise, your muscle was looking heavy, bulky. That is because of the lactic acid accumulation. Till the time in the evening, these lactic acid molecules will slowly will drain out from here, and again your muscle will almost look like the normal one which was earlier. These lactic acid molecule will reach all the way to liver. Now what the liver will do with this lactic acid? The liver will convert this lactic acid back into the pyruvate. And to convert that into pyruvate, what we need to do is we need to use an enzyme that is called as lactate dehydrogenase. With the help of enzyme lactate dehydrogenase, lactic acid will convert into pyruvate. And that pyruvate, we know that just now we have discussed, the pyruvate can be converted into glucose. Just now we have discussed about that. that how the pyruvate will convert into glucose, that will go inside, will make oxaloacetate, and then we know how to go back up all the way. So this is how the lactic acid can ascend up and can reach all the way till glucose, right? So this is called as the gluconeogenesis that is going to occur with the lactic acid. And once this glucose is made, this glucose can again be sent back all the way to muscle for the energy purpose. This glucose molecule can be sent back all the way to the muscle for the energy purpose. And this entire cycle is referred as the coA cycle. This is referred as the coA cycle, right? So this yellow color arrow is the point of discussion for right now, that how the glucose is to be made from the lactic acid. But my agenda was to make you understand that from where this lactic acid is coming, right? The reaction part is not very important. You can see you just have to remember one reaction: lactic acid converts into pyruvate via the lactate dehydrogenase. The rest of the thing we already discussed, the how to make glucose from pyruvate. So this is how the coA cycle occurs, and this is how we can generate the glucose from the lactic acid in the liver. Let's see the third substrate. The third substrate is the amino acid to glucose. How we can convert various amino acid into glucose. How we can convert various amino acid into glucose. Various amino acid into glucose. So I will start with the first amino acid, that is tyrosine. If you know that tyrosine, when we have discussed the session of tyrosine, we have discussed that tyrosine is required to make three special products. What are the three special products of tyrosine? Melanin, T3T4 and the catecholamines. Melanin, T3T4 and the catecholamines. If all the three are there in the normal quantity, then what is going to happen with the tyrosine? Then this tyrosine is going to be catabolized. And the pathway, the various intermediate of the tyrosine catabolism pathway is, it will convert into 4-hydroxyphenyl pyruvate. We have discussed that in detail in the tyrosine session. Then it will converts into homogenetic acid. Then it converts into mallyl acetoacetate, the fumaryl acetoacetate, and ultimately it will convert into fumarate and acetoacetate. This is the tyrosine catabolism pathway. The mnemonic that I have told you how to remember is the sequence of event when you do alcohol party. हम पहले पीते हैं, first we drink, then what we do is हम गाते हैं, that is singing, then what we do is the mother will come into the picture, then the father will come into the picture, and ultimately is the fumarate. Yes, that was the mnemonic that we have used to remember the tyrosine catabolism pathway. Now today we have in the session of the Krebs cycle, you may have seen that one of the intermediate of the Krebs cycle was fumarate, right? So fumarate can convert into malate, malate can convert into oxaloacetate, right? So I can say that fumarate can easily be converted into oxaloacetate because both are the intermediate of the Krebs cycle. Fumarate can easily be converted into oxaloacetate via the TCA cycle. You will be using the enzymes of the TCA cycle, and the fumarate can convert into oxaloacetate. And just now we have discussed that oxaloacetate, once you have made, you know how to ascend up from the oxaloacetate and reach all the way till glucose. When we were making glucose from the pyruvate, we have seen that if you have oxaloacetate, that oxaloacetate can easily go all the way up till the glucose. This is how we can make glucose from tyrosine. In the session of tyrosine, we have discussed that tyrosine is a non-essential amino acid. The reason why it is non-essential amino acid, because it can be synthesized from an amino acid that is with the name of phenylalanine. So if I want to make glucose from phenylalanine, is it possible? Yes, it is possible. Phenylalanine can convert into tyrosine, and tyrosine can convert into fumarate, oxaloacetate and glucose. This can be done. It is easy and straightforward. Then if I want to say aspartate, can we convert aspartate into glucose? Yes, it is possible. Aspartate can convert into oxaloacetate, and the name of that reaction is transamination. That we have discussed when we have discussed the biosynthesis of urea. Transamination. The mnemonic for the transamination that we have used in that session was the SOLP. S stands for aspartate and O stands for oxaloacetate. So when you do the transamination, S aspartate, O is oxaloacetate. So you will get the oxaloacetate, and for oxaloacetate you can make glucose. In the same way, if I say, let's say there is one more amino acid, alanine, and from alanine I want to make glucose, yes, it is possible. It is possible. You will do the transamination, you will get the pyruvate, and from pyruvate we know can convert into oxaloacetate and we can make glucose. The mnemonic was LP. L stands for alanine and P stands for pyruvate. So the mnemonic for transamination that we have used was SOLP. S aspartate, O is oxaloacetate, L is alanine and P is pyruvate. This is how we can make the glucose from the various amino acid. Let's add few more amino acid in this list. We know that valine, isoleucine, methionine and threonine. These amino acid can make an intermediate of the Krebs cycle with the name of succinyl CoA. They can make an intermediate of the Krebs cycle with the name of succinyl CoA. If you can make any intermediate of the Krebs cycle, that succinyl CoA can easily be converted into oxaloacetate via the TCA cycle. Via the TCA cycle, we will be using the enzymes of the TCA cycle, and this succinyl CoA is going to be converted into oxaloacetate, and that will convert into glucose. So the basic idea is that we want to make an intermediate of the Krebs cycle. If you are able to make an intermediate of the Krebs cycle, from that you can make oxaloacetate, and we know that from oxaloacetate how to make glucose. This is what we have discussed earlier, that from once you have made the oxaloacetate, how to ascend up and how to go back all the way up to glucose. So this is how we can convert the various amino acid into glucose, and the most glucogenic amino acid is alanine. This is the main substrate of gluconeogenesis. The major substrate of gluconeogenesis is amino acid, and in the amino acid, if they ask which one, it is the alanine. Alanine is the main substrate for gluconeogenesis. Then let's see the fourth one. The fourth one is from the oat chain fatty acid how we can make glucose. Oat chain fatty acid to glucose. How we can convert the oat chain fatty acid into glucose. To convert the oat chain fatty acid into glucose, let's see the oat chain fatty acid will convert into, when you do the beta oxidation, at the end of the beta oxidation of oat chain fatty acid, there is a product that is formed, and that is called as propionyl CoA. Propionyl CoA is a 3-carbon molecule. This propionyl CoA can be converted into methyl malonyl CoA, which is a 4-carbon molecule. 3 carbon converting into 4 carbon, so the name of the enzyme is propionyl CoA carboxylase. And for that we will be requiring vitamin B7 because it is a carboxylation process, so requiring B7. Then this methyl malonyl CoA can convert into succinyl CoA, which is a 4-carbon molecule. Methyl malonyl 4 carbon, succinyl CoA 4 carbon. The name of the enzyme is methyl malonyl CoA mutase. And we require vitamin B12 for this. Now once you have made the intermediate of the Krebs cycle, this is an intermediate of the Krebs cycle, succinyl CoA. Once we have made an intermediate of the Krebs cycle, from that we can easily make the oxaloacetate via the TCA cycle enzymes. Via the TCA cycle enzyme we can make the oxaloacetate, and from the oxaloacetate we know how to make glucose. Right? So this is how we can convert a oat chain fatty acid into glucose. What is the clinical correlation that we should be familiar with, and that is really highly tested. See, what they are going to give you is, they are going to give you a scenario that, first let me make you a background clear that there is something called as macrocytic anemia. What is macrocytic anemia stands for? Macrocytic anemia means the MCV, the mean corpuscle volume, is going to be more than 100 femtoliter. It is going to be more than 100. If the MCV is more than 100, we will say it is macrocytic anemia. If the MCV is more than 100 and hemoglobin is obviously less, if the hemoglobin is coming in the range of anemia and the MCV is more, we'll say it is macrocytic anemia. Macrocytic anemia. If the MCV is less than 70, we'll say it is microcytic anemia. So if the MCV is more and the hemoglobin is less, we'll say it is macrocytic anemia. Now there may be many causes of that, but what we are concerned with, it can happen with vitamin B12 deficiency, it can happen with vitamin B9 deficiency. It can happen with vitamin B9 and B12 deficiency. Right? So let's say there is a patient comes to you and he is anemic, he is having pallor, the palmar creases is not visible, he is having shortness of breath on exertion and he is looking pale, right? He is anemic. Right, just looking in the OPD, we have made a diagnosis anemia. Now we have to figure out that what is the cause of anemia. So you ordered a CBC, complete blood count. In the complete blood count, the hemoglobin comes out to be, let's say, 6. That is anemia, and the MCV comes out to be, let's say, 120. MCV comes out to be 120. Means it is a macrocytic anemia. Now the probable causes can be one is the B12 deficiency, or it can be B9 deficiency. It can be because of B12 deficiency or can be because of B9 deficiency. Both can lead to macrocytic anemia. Now, we need to figure out whether it is due to B9 or B12 deficiency. So, what is the next thing that you will do? Then the next thing that you are going to do is you will check the level of methyl malonyl coA. You will check the level of methyl malonyl coA. Now, why we check the methyl malonyl coA level? Now, see here. In this reaction part, what you can appreciate here is if the B12 level is less, if there is B12 deficiency, then this reaction is not going to happen. If the B12 is less, this reaction is not going to happen. This is not going to happen if the B12 is less. Then what is going to happen with the level of methyl malonyl coA? The methyl malonyl coA is going to be raised. If it is B12 deficiency, methyl malonyl CoA is going to be high. If it is B9 deficiency, it is going to be normal, right? So to differentiate a patient of macrocytic anemia, whether it is a B12 deficiency or B9 deficiency, what you will do is you will check the level of methyl malonyl CoA. If the methyl malonyl CoA level is raised, then you will say it is vitamin B12 deficiency, because this reaction is not going to happen and the methyl malonyl CoA will accumulate. And if the methyl malonyl CoA level is normal, then it can be because of vitamin B9 deficiency. So this is very highly tested, that they will give you a clinical scenario, and based on that they will ask that whether the anemia is because of B12 deficiency or B9 deficiency, and we have to figure out that based on the given lab values whether it is B12 or B9. So you check the level of methyl malonyl CoA. If it is raised, it is going to be B12 deficiency. If it is raised, it is the B12 deficiency. So this is how we can make the glucose from the oat chain fatty acid. Let's go to the next one, that is the last substrate for gluconeogenesis, and that is with the name of glycerol. How we can convert the glycerol into glucose? So to understand this, what we can say is, see, the glycerol converts into something called as glycerol 3-phosphate. And how it will convert into glycerol 3-phosphate? We will bring ATP into the picture, convert into ADP, and will transfer the phosphate on the third carbon of the glycerol. So because we are transferring the phosphate, so the name of enzyme is going to be glycerol kinase. Then this glycerol 3-phosphate will convert into something called as dihydroxy acetone phosphate. Now to convert the glycerol 3-phosphate, so glycerol 3-phosphate will convert into dihydroxy acetone phosphate. Now to convert it, what happens is, in this reaction, NAD comes into action and converts into NADH. And we know that whenever this NAD, NADH, the name of the enzyme is going to be dehydrogenase. So it is going to be glycerol 3-phosphate dehydrogenase. Then this dihydroxy acetone phosphate is going to convert into its isomer, that is glyceraldehyde 3-phosphate. because they are the isomer of one another. So the name of enzyme is isomerase. Dihydroxyacetone phosphate and the glyceraldehyde 3-phosphate both are of 3 carbons, right? So they are the monosaccharides C3H6O3 O3. So the same formula, but the enzyme molecules are different, so they are the isomers. Now once you have made this glyceraldehyde 3-phosphate, this is an intermediate of glycolysis. If you remember, this is an intermediate of glycolysis. Now if you can make any intermediate of glycolysis, you know that how to ascend up in the glycolysis. Whatever irreversible step that is you are going to face, we know that alternate routes, and we will ultimately reach all the way till glucose. We will reach all the way till glucose. So the point is, what they ask is, when the glycerol enters in the gluconeogenesis pathway, it enters via which intermediate? It enters via the glyceraldehyde 3-phosphate. It enters in the gluconeogenesis pathway via the glyceraldehyde 3-phosphate. First it converts into that, and then we know that once it is made, it can easily ascend up and it can easily go all the way till glucose. That we have discussed when we were discussing from pyruvate to glucose. How to ascend from pyruvate to glucose. There were 3 hurdles. So in between, the glyceraldehyde 3-phosphate is an intermediate, so you can enter that pathway and you can ascend up all the way. Right? So this is how we can make glucose from the various substrate, maybe pyruvate, lactate, amino acid, or the fatty acid, and lastly is the glycerol. You can make glucose from many submolecules, but we can never ever make a glucose molecule from the acetyl CoA. We can never ever make. Now, having said that, let's see the regulation part of the regulation of gluconeogenesis. Regulation of gluconeogenesis. So what we are doing in gluconeogenesis, the non-carbohydrate molecules are going to convert into glucose. And this process is referred as gluconeogenesis. This process is referred as gluconeogenesis. So now we have to see that who is going to stimulate it and who is going to inhibit it. To find out, we need to see this in respect of insulin and in respect of glucagon. My question is, what is the product? You need to see the product. The product is glucose. Insulin wants to make the glucose or wants to decrease the glucose. Insulin does not want to make glucose anymore. So gluconeogenesis is going to be inhibited by insulin. Whereas the opposite, glucagon wants to make more and more glucose, so it is going to stimulate it. So once you know the concept, then it is going to be very easy, right? Now let's see what will be the effect of acetyl CoA. I am saying that the acetyl CoA is never ever a substrate of gluconeogenesis, but now what I am saying is how it is going to affect the gluconeogenesis. If you increase the amount of acetyl CoA, is it going to stimulate or not? It is not a substrate, that is fine, but it is going to stimulate the process or it is going to inhibit the process, right? So let's understand that via a simple physiology diagram. I can say, let's see here, and then we will apply the same concept on the acetyl CoA. For example, there is a molecule A. This A molecule can convert into B. For that, the enzyme is, let's say, Z. The name of the enzyme is Z. If you increase the concentration of B, means if the B is already there, what it will do is it will give a negative signal that I am already there, no need to convert A into B, I am already there. Right? It will give a negative feedback. This is called as negative feedback phenomena, that is usually done by the products. Whatever is product, if the product is already there, it is in high amount, it is going to inhibit the enzyme. Now, let's say that this A can also convert into a molecule that is with the name of C, and for that the enzyme is D. So what if the increased concentration of B, what it will do? It is going to inhibit its own enzyme. That is fine, that is very straightforward, but what else it can do is, it can stimulate this enzyme. It can stimulate the D enzyme. If it stimulates the D enzyme, then what is going to happen is, the A will be diverted more towards the C. And this is what the B wants. If the B is already there in the higher amount, what it will do is, it will inhibit its own enzyme and it will facilitate the other way, so that A will be diverted towards the D side and the B will not be made anymore. So if I use the same concept here, there is a molecule called as pyruvate. There is the same molecule called as pyruvate. We are saying that the pyruvate can convert into acetyl CoA, that is the link reaction, if you remember, link reaction. Pyruvate can convert into acetyl CoA. For that reaction, the name of the enzyme is PDH complex. PDH complex is the enzyme for that reaction. So if I increase the concentration of acetyl CoA, it is going to inhibit the PDH complex. Once it is there, it will inhibit that, no need to make me anymore. The pyruvate, just now we have discussed that pyruvate can be converted into glucose. And that process, pyruvate to glucose, is referred as gluconeogenesis. So if you increase the concentration of acetyl CoA, it is going to inhibit its own enzyme. That is very good. But at the same time, what it can do is it can stimulate the gluconeogenesis. So more pyruvate will be diverted towards this side and will make more glucose. So acetyl CoA is never ever a substrate of gluconeogenesis, but it can stimulate the gluconeogenesis. It is never ever a substrate, but it can stimulate the gluconeogenesis. So this was the concept that you need to understand by this diagram. I hope you have understood this concept. If you understood, very good. If not, you need to remember it directly that acetyl CoA is not a substrate, but it will stimulate the gluconeogenesis. It will stimulate the gluconeogenesis. This is the point that you need to remember. Okay. Thank you, guys.